So today we are going to discuss about genomic DNA libraries. Okay, and we are mostly going to compare this with that of cDNA libraries. And today we are only trying to see the aspects. We're going to continue with what we have learned in the previous class. We in the previous lessons we learned about vectors. In that I have already introduced to you how a lambda phage can be used, and the same thing check on that. One second, there is. So in the previous ones, we have learned about restriction digestion, a lot of other cloning strategies. And now, say for example, why do we need to go for genomic libraries? Using PCR could be huge, especially if you take that of higher organisms or complex, sometimes much larger, because of the presence of introns. Okay. So what happens is that we will not be, if they are larger than something like this, if they're more than 5 kb, then there are going to be mistakes, especially if you're any tag DNA polymerase or so. If you're using, there are errors that could happen while the genome is something else. Once it could have had a different sequence, and when you clone it, because tag DNA polymerase might make a mistake, you will have some errors because of that. Proofreading activity of tag DNA polymerase. There are also in some cases because of premature termination. That is, maybe it's not always that the whole amplicon is the say for example it is like that. Sometimes we may have only partial sequences cloned when you do ligation or anything, and because of which we are not getting the absolute information of that genome, right? And that is one of the limitations using PCR based ones. There are other, as in you can use long range PCRs. That is like you use two different types of polymerases. One that has high processivity or speed, such as tag DNA polymerase, and some that are some of them, and also another DNA polymerase which is having high fidelity, such as PFU polymerase. Okay, but even then we have these are possibilities that we could do. If the gene is too long, we can use these kinds of techniques. But I'm using it in the context of genomic library about what are the limitations of PCR and other things. And there are also other remedies, or you can say solutions, that are given. One could always try to use those. So for a genomic library, the first thing, there are several steps involved. That is to first obtain genomic DNA and then break it into, because the genome is huge, we need to cut it into fragments, and that is usually done by restriction digestion or mechanical shearing. Okay, you need to give me one small. Then the second one is to ligate it into all the fragments into a suitable vector. We have learned several vectors, of which for genetic libraries or genomic libraries, we used lambda EMBL3A. If anybody has a question, please you unmute your mic and speak up the question so that I can see that you're having. Otherwise, I'm using single monitor, so I cannot see your messages or anything. And the second one is then you will be able to create a library and after that, we should be able to screen the library. Screening the library is finding what we want, finding the gene of interest or clone of interest. Right? When you have taken the whole genome, broken into pieces, and all the genes could be, or all DNA fragments that are representative of the genome are inserted, and we need to find that particular clone that we are interested in. So there are several techniques for screening. We will learn that in the later classes. Once we have done that, we should amplify our target sequence, and then further characterization can be done. So I hope you remember this figure. We already used it twice, I guess. So this is the third time that we are doing. And now the focus is about the genomic DNA. In all the previous times, our focus was as a vector. Okay? Remember the objective always. The objective here is to have, say, for example, we have a genome and it has several genes A, B, C, D. And what is a genomic library? We have to first define that. A genomic library is a set of clones which has all the gene sets represented at least once. Right? Now you have 4 clones. Each of them is having A is represented, B is represented, C is represented, and also D is represented. If you had a set of clones, something like this, you can have, say, 8 of those. And now I'm going to have genes such as A, B, D, B, A, and D. Is this a genomic library? It is not a complete genomic library because C is missing. Right? So if one wants to define a genomic library, it should have had it's a set of clones which have all parts of a genome represented at least once. Say if you also have another one here, and now it has a C. Although B, A, D are represented twice and C is represented only once, it is still all right. It is a complete genomic library. Okay? So our job is we have the genomic DNA, we have isolated it from an organism, and now we should ensure that each part of this genome are made into a clone. That is, it should be in a lambda vector, it could be a plasmid, or an yeast artificial chromosome or bacterial artificial chromosome. Here we are using lambda-based vector, that is lambda EMBL3A, as a vector for constructing genomic library. So we will just go through the steps, and this should be a revision of what we already learned in the genomic libraries. Okay. Am I audible? Somebody?
Yes, sir.
Okay, good. So the first thing is we will take the high molecular weight genomic DNA because it's usually huge or large amount. The length of it is huge. We will try to use partial digestion using South3A. Why did we choose South3A? South3A A1 is I think its sequence is GATC. That is the site it recognizes and it cuts, and it is a 4 base cutter. So 4 into 4 into 4 into 4, I think it's around 1,000 bases or so, 1 kb. It cuts about, is that right or am I wrong? 16. Can someone confirm? Or is it 256? Yes, it cuts about every 256 bases. And remember, it is a partial digestion. It's not complete digestion. So why are we using something like partial digestion? We are going to have a gene like this. Say, for example, there are 3 genes on the genome or a chromosome, and this one is about 1 kb. This could be about 2 kb, or this could be about 3 kb. And if it cuts every 256 bases, and if you allow complete digestion, each of it would have been broken into small fragments. And then reconstructing back what we want is very difficult. So there are 2 ways of doing this. One thing is one could have used a restriction enzyme that cuts rarely, or maybe 6-base cutter is fine. Sometimes 8-base cutter is also fine. It depends on your objective. But for something like when we are using or planned a construction of a genomic library based on the lambda vector, we would want a little more construction. We will see why we need that. Say, for example, I allowed partial. So in one case, it might be cut might be here, here, and here. So what do I get? I'll get a fragment like this. In a different genome, I mean, a different piece of the same DNA, it might be cut differently here and here like that. So in that case, I would get a DNA like this. We are actually generating overlapping fragments because of partial DNA digestion. And we are using a frequent cutter, or it is a 4-base cutter, so it cuts DNA frequently. And at one point, if we have generated these, we will again discuss this when we are talking about library sequencing, shotgun sequencing, and other things. But if we have a clone of each, and now when we kind of accumulate, we should be able to read the whole genome as a continuous one, because there are overlapping ones here. We will also learn some techniques of something like chromosome walking, which you may have learned. It's an ancient technique in a way for the current technique, for the modern technology that we have, but it is still interesting. So we do a partial digestion with a 4-base cutter for this reason. And then we are treating with phosphatase. I hope you remember that we always maintain that typically we treat the vector with phosphatase, because we want to avoid religation. In this particular aspect of genomic library, we want to prevent any ligation between 2 fragments like this. Say, for example, you cut it with South3A, and then you have treated with ligase. Okay, in the ligation reaction, what may happen is I will call this fragment A, this fragment B, and this fragment C. And when it is constructed into it is cloned or ligated into vector, these 2 might ligate and then ligate with the vector A and B. For this, if this is the case, I don't have any problem, because they were the original genetic architecture on the genome is that these fragments were side by side. What if something like this happens, where fragment A is ligating with C, and then I get a fragment like this. When upon cloning, upon ligation, it also A ligated with C, and A and C fragment ligated with the vector. Now we have a confusion. Which one is the correct scenario in the genome? My job of trying to construct the genomic library is to also study about the order of genes, any intergenic regions, and all things. We are trying to read it as a genome. We are not trying to read something as a gene. The primary objective of genomic library, maybe 2 decades early, was to sequence the whole genomes of bacteria or any mammal or human even. And in that case, I'm trying to say, for example, trying to know the genome, the order of sequence, order of genes on a human genome. And if I get a scenario like this by serendipity, it is all right. But if I get a sequence like this, it is actually confusing me whether B is beside A or C is beside A. We cannot say that. So we need to somehow prevent the real ligation between or among the fragments that are produced. Therefore, they treat the fragment with the alkaline phosphatase. So alkaline phosphatase will remove the phosphate group, and then that once it is removed, now we are preventing the possibility of A ligating with C or A ligating with B. The only possibility is it can only ligate with the vector. So we are likely to then, upon ligation, we will get only A on one, B on one, and C on one. Of course, if you want to work out the order of genes, we are going to have these overlapping fragments in some cases. When we read about chromosome walking, I will discuss that part at that part in that region or in that topic. So once we have done that, we will ligate with the vector. And I will just quickly go through vector. You can also try to think of it along with me. Sal1, BAMH1, EcoR1. And the reverse order is on the other polylinker. EcoR1, BAMH1, Sal1. And there are also two genes, red and gam. I hope you remember or you can recollect it. Red and gam are something involved with spy phenotype. If red and gam are present, it is spy positive. If red and gam are not present, it is spy negative. What is spy? That is sensitivity to P2 inhibition. Okay, that means if there is a lysogen, that is, I'll draw here, there is a bacterial cell, it has a genome, and on the genome you have P2. That is a prophage of a P2 phage. If it is if this is a lysogen and the genome of it, the lambda phage somehow cannot infect P2 lysogen, and the molecular determinants of that sensitivity to P2 inhibition are red and gam. In the red and gam, you will have, if we delete this red and gam, say lambda is present and it is red and gam minus, then it can infect the P2. 1 second. Sorry. Okay, that is about why they included red and gam. You should also remember that lambda EMBL3A is a insertion vector. It is a replacement vector. That means it is about deleting some part of this stuffer region and incorporating the fragment of interest, such as this one. So EMBL3A is digested with BAMH1 and EcoR1. So we are using two enzymes to cut it something like this, BAMH1 and EcoR1. So there are three big fragments: left arm, right arm, and the stuffer region. You also have these polylinkers. You can simply do isopropanol precipitation and throw away the supernatant, which is likely to have. You are eliminating these polylinkers, because of which the left arm cannot. We are preventing the stuffer region from religating with either left or right, because as you might notice, stuffer region has EcoR1 overhangs, whereas right and left arms have BAMH1 overhangs. Because we eliminated the polylinkers, the stuffer region can no longer ligate with left arm or right arm. And then we will now ligate these two, use these three large fragments, that is left arm, stuffer region, and right arm, and then add also the fragments, the DNA that we are looking at, and then we will do ligation. When we perform ligation, there are several possibilities. I would encourage you to go to the video where I have described those in detail. But what is simple way is those that have stuffer region cannot ligate. Even if it ligates, it is eliminated in the because we are using a P2 lysogen. And only those that do not have left arm will not form a plaque, because there are essential genes that are required. If left arm directly ligates with right arm, then they have all the essential genes to form the lambda phage, but their size 9 plus 20, I guess somewhere around 29 kb, which is not good enough. I think 9 plus 12 or something should come, I think. Either way, they will fail to form infective phages during in vitro packaging. So how we are increasing that only the recombinant with the fragment are formed, left arm fragment and right arm, is using several aspects. First thing is we are using two restriction endonucleases and eliminating the polylinkers, as we have shown these ones. Second thing is we have included red and gam. So if there is any case of stuffer region getting incorporated, those will get eliminated during selection on P2 lysogen. And if there are any that do not get the fragment, those are eliminated during in vitro packaging. So that is how we will ensure that we will actually get a large number of plaques. Each of those is each of that lambda is having one gene like this. Okay, within the lambda it is linear. So, but anyway, just assume that these are, each one is in one of the lambda like this. That is how one constructs a genomic library. I mean, there is little more calculations to be done, Carbon and Clark formula. Maybe we'll discuss the fundamentals of it in the next class. Okay. So one needs to generate a large number of clones to ensure that each of the genetic elements is represented at least once. If any one of the genetic element is missing, that is not represented in that clone, then it is not a complete genomic library. But when we really do things in the lab, there are usually a lot of difficulties. We cannot say everything with certainty. So sometimes we have to use statistics and probability with which one can say that every genome is represented with 95% probability or 99% probability. Okay. We will also see one more aspect. That is, I like this one too for one reason. This is called as lambda Charon 4a. This is also a lambda-based vector. It is a replacement vector. So in this case, we take high molecular weight DNA, and we are cleaving with a mixture of HAE3 and ALU1. And you're doing partial digest for the same reason as what we are doing before. And then you do size fractionation. What is size fractionation? Pulse field gel electrophoresis. I hope you know this one, where you have alternating currents. Maybe in the next class we will have a picture of it, and we'll try to discuss that. Upon digestion, you may have several fragments like this, several sizes, right? Because you're doing partial digestion. Some parts might be digested more, some parts might be digested less. In some cases, you may have these restriction sites in higher concentration within a region. Some of them may not have these sequences. So we need to, if we want to have a, um, we need to sometimes have some certainty where, if for calculation purposes at least, is if we have some 20 kb fragments, then we should know, we would be able to calculate how many fragments, how many, what should be the size of the genomic library. Okay. So in that case, you can also think of it as this one, where you have to do electrophoresis, and then all of these will get fractionated based on, I mean, they based on the size they travel. The smaller will travel farthest, and the larger will travel slower. And where it comes to around 20 kb, you need to cut that part out and then use it for in your analysis. Okay. And there is one more thing they do before, once upon size fractionation. I want to emphasize this one part where they use EcoR1 methylase to block all the EcoR1 sites around. If there is, if you now you have all the fragments that are approximately 20 kb, and you are supposed to clone it in EcoR1 site. Okay. And if we want to use EcoR1, it's not necessary that these ends are not EcoR1 type. They are, we have used different other and restriction endonucleases. So we need to attach either linkers or adapters. For this particular protocol, they use linkers. Linkers are the ones that have the restriction site, something like EcoR1 like this here, which should then be treated with EcoR1 to get the overhangs. But what if the sequence that we have also has EcoR1? Then it is going to cleave that part, and that's not going to be a good thing for us. So what they have used EcoR1 methylase to protect all the EcoR1 sites that are present on the fragment, and then attach the linkers. Okay. And then you digest it with EcoR1 and go for ligation. Okay. So alternately, we can use adapters also, but I think working with linkers is easier, provided you have a scenario like this. Adapters could be sometimes tricky in using them. So on the lambda, on the vector side, we have the cohesive ends. You can anneal them because the cross sites are here. And now you can treat it with EcoR1. There are 3 EcoR1 sites. And now you have a linear part. And these are the 2 internal fragments. You size fractionate, same thing as using pulse field gel electrophoresis also. When you do size fractionation, the larger fragment will be there and the smaller ones will be here. So you just cut this piece out and you isolate the DNA. So it is a replacement vector. We are removing these regions and putting the gene of interest in that location. Now you can, once that is over, we mix it along with the processed fragments, and then we add ligase. Once we had ligase, there are multiple possibilities, out of which one of it would be like adding, this is a cost site, right? This is another cost site. This would be another cost site. So this, it might form a molecule of repeating units like this, and that is called as a concatemer. I hope you remember this concatemer when we discussed about the lambda phage biology. From once it is internalized during infection, it circularizes. And from circularized double-stranded DNA, it undergoes theta mode of replication. something like this. This is to say this is one origin of replication that is going this way. This is another origin of replication. From here, it shifts to rolling circle model of replication, and it keeps on producing concatenamers. This is one replication fork, and it keeps producing a long stretch of repeating lambda units, and that is a concatenamer. So when we are doing it with in here in vitro, when we add ligase, there is also a possibility that they can form concatenamers like that. That concatenamers is a good substrate for phage packaging, in vitro packaging. So now you also should have made the head particles and the tail and other things outside using a helper phage or helper cells. You make those, you produce the structural proteins of the phage from other cells. And in vitro packaging, you add the ligation mix like this and the head particles. Then you will eventually get phage particles loaded. So this cos site will be recognized, and the rest of the DNA will be pushed into the head until the next cos site is arrived. I mean, once the second cos site is there, a cut is made and the packaging is completed. Okay. So this is about two methods of using constructing genomic libraries using lambda phages, lambda phage based vectors. So there are many other vectors that can be used, especially these will be much bigger, while lambda phage has a limitation of about 20 kb or so. BAX and YAX can go to, I think Cosmids has about 50 kb or something like that, 45 kb. BAX and AX can go much higher, 100 kb and above. Okay, this is something around 45 kb. So why are we doing genomic libraries? The first thing is to determine the complete genome sequence. Now things have become much easier. We don't have to go through the process of constructing genomic libraries. People directly take the DNA and go for sequencing, next-gen sequencing, which is good, which is highly informative, accurate as well, but still it is very expensive for a typical lab to afford. But still, these are good strategies of how one can engineer genetic elements to achieve a goal, and that's what these are. That's why genomic libraries are fantastic things to learn. We will know how flexible our thoughts and approach could be when we want to use genetic elements for our purposes. That is the objective of this course, is to empower you with the flexibility of thought and the ability to you should know all the genetic elements and the way one can use them. That ability is what I hope you are all gaining from this course. Then it is also, if we are cloning genes, we might not necessarily know about several regulatory sequences, right? If we are just looking at ORFs or tRNA genes or non-coding genes, what about the regulatory sequences? It would become very difficult to learn one gene. Okay, I have gene A. What are all the regulatory elements here? Gene B. What are the regulatory elements here? Something like that. We are missing a lot of information about the intergenic regions, right? Many of them used to call, because they are large, used to call them as junk DNA. Even the introns are also sometimes caused. But with the increasing knowledge that we have right now, we are thinking that many regions that were thought to be junk seem to have some kind of function. They might encode small RNAs and other things. So if it isn't for people constructing genomic libraries and trying to determine the complete genome sequence, the importance of this intergenic region was not known. Only through genomic libraries, we used to know these things, the existence of them in the first place, right? These are all majority of these techniques are before the advent of next gen sequencing. If I will also at this point will also tell you people, if you are trying to become a hardcore researcher, I would suggest you to be very aware or get to a level of biological data scientist, that is about managing uh, all the sequence, right? So one should be good with two things. One is the next gen sequencing, because that is the future. In future, we will have more data and less number of people working on that. Second thing is to ability your programming skills. Okay, programming to manage the huge amount of data, and that data is biological data here. Of, of course, it is needless to say, if you are good with this or good with this, it is all right, but you should know the biology very well. Okay, biology, your base is biology, and one can become a specialist in these. Even if not a specialist, one should be able to write small algorithms for analyzing sequences, small ones. Especially why I'm saying is, in the last time when we were looking into some of the openings, majority of the openings say people with ability to write small algorithms are preferred. So in any kind of biology that you might be doing in the future, you will have to do these. So if I'm sure you already know programming. It's not a big deal for you because you already learned. So I don't know programming. I haven't learned it ever. I'm from a pure science. So I have difficulty with this particular part at this point, like this. I'm trying to learn. That's a different issue. But if you already have an initiation into programming, so I would suggest you to take up some courses on Coursera or so, and try to add this to your CV, so that you are empowered to analyze large sequences. Okay. So I will stop here. In the next class, I will take cDNA library, and I would want to know about if you have any questions, and also I want to know if you had any questions from the previous class. Those are the two things that I would want to know. Please, you can switch on your mic and let me know the questions that you have. Sorry. Any questions? Or even if today's class is also fine, you can ask me questions.
Sir, I have a question from previous lecture, sir.
Yes, tell me.
Sir, about the replica plate, from one master plate, how many replica plates can we make, sir?
1 second. So this is from PBR 322,
Yes,
right?
sir. Yes, sir.
Okay, you are having replica. You can make 3 or 4 replicas, but each time it's when we are discussing on paper for teaching purpose, we'll just put 4 or 5, right? And you can make replicas. That's not a big deal. But if you have say 100 colonies and you're trying to do multiple replicas, sometimes there might be an error. Actually, I tried to do this in my lab too. It's not that easy actually.
So
But
then why do they prefer to do this method, sir?
no, at that time it was preferred. Now we don't do that. We actually take a colony. Okay, I will, if I have to do this now, if I have some 5 or 6 or say even 100, it is laborious process to do in this way. What I would do is pick a colony, make a patch of it like this on 3 different plates. Second colony, I should have a patch like this. Okay, each of these plates is without any antibiotic or with antibiotic. Okay, and I could have made a patching like this for all the sequences or all the colonies that are here. But this is so laborious because if it is 100 colonies, then I have to do 100 and count into 3. That's the number of times I have to make these patches on these plates. If it were replica plating, one cloth, and then I think some people have stabilized it or did better. We tried it without velvet cloth and other things, which is not a good way of doing things. In those days, I think they have managed it. Once you find a colony or not, people again streak it and verify whether that is true or false. Okay,
Okay,
this
sir.
is just a screening technique, but you can do it 3 or 4. I think it should not be a problem because what you have on the velvet, even if suppose when you put the print and took it out, there were assume in this colony there were 10 to the power say 1000 cells, and here in that place only 10 cells have uh, are bound, okay, are stuck to this velvet. Even then, if you get 2 cells here, 1 cell here, that is fine enough. It will form a colony, no problems there.
Okay, sir.
Okay,
Yes,
it's
sir.
only whether we are making the imprint properly or not. Say sometimes if we put it slant and say this region didn't get the imprints, then we might not have a colony.
Okay, sir.
Yes. Any other questions? Any questions from the last class? I thought that is what I was going to. With the imprint, people should be having some issues. Yes, no? In when you learned about when you learned stem cell biology, did you learn about genomic imprinting? Is there anyone who has taken that elective from biotech?
Yes, sir, I took, but we didn't do anything on genomic imprinting, sir.
Okay, but that's okay. That's all right. So it's per se, it is not given in the syllabus as such, but the techniques that were discussed are actually based on genomic imprinting in stem cell biology. In case of here, we have to learn about, we are using a technique to study epigenetics, and that to study imprinting. If you want, I mean, once I make a video, I will try to upload, and then you can probably watch it, and then you may have questions. There is also one more thing I wanted to say. Please note down a few techniques, and I would want you to read for the next class. For the next class, we will have cDNA libraries. Okay. We will discuss about cDNA libraries. I would want you to please read about it and come. There is also about sequencing. There is one more method used for studying the epigenetics. It is bisulfite sequencing. Read about it and come. Okay, these two things I would want you to have a look at it before you come. This bisulfite sequencing I will not teach until an assessment is made. An assignment will be given later, and then we'll based on that. Okay, any other
Sir,
questions?
I have one more question, sir.
Yes, please.
Sir, in the EMBL3A, in the video you have discussed various
Yes.
possibilities of how the left and right fragment will get ligated and how we will screen it out, right, sir? In that, what
That's
if
right.
a left fragment is combined with a right and then a left? It makes a size of about like 15 or 49 or something, which will come under the packaging size, no, sir. In that case, how will we eliminate that?
Um, I think I will try to answer rather. Okay, sorry. Um, I'll go here. So you mean to say we have a left, sorry, left, and we have a right, and another left has like it.
Yes, sir.
Is that right? So the size is something 9, 20, and 9, something
20,
like that.
9, and
So you'll
20,
get the
sir.
size is within the range. Yes?
Left is around 20 kb, sir. Right is 9, sir.
Okay. Okay, okay. My bad. 920 like this. So you have
40. Yes.
49. So size wise, it is getting packaged.
Yes, sir.
So first thing is the left and right, when they chose, they will have this is compatible with this right, right?
Yes, sir.
I think assume that this is eco R1 and this is eco R1. Just assume. And these sides, I think it is usually blocked.
Okay, sir.
And this should be, if they like it should be a cos site. If they like it like this is one, here is one cos site, okay? If it
Oh,
has formed a multimer like this should have been a cos site too.
okay, sir.
During packaging, this is the other end, right? Because the end, this, sorry, how do I say? Uh, the end here is cos, and this end is cos. So when they ligate between these two, then you form a L and R like this. When they ligate this way, on the other side, there is a cos site that is forming. So the size of it will still remain around 29.
Okay, sir.
So it doesn't matter if this one has formed also. It will still count this one only.
Okay, sir.
Even if it assume that this one is formed and by mistake and so on. When it is forming a concatemer, what would happen? What would be the size? Right? And then again, you will have cos site, cos site to cos site is 29, 29 kb. So new phages should form, isn't it? Plaque forms, not just infection is not enough. If it is infection, then you have replication to happen. Then you have the new phages to form. These phages should break open the cell and come out. Lysis will happen then. So infection, replication may happen, but during packaging, again, 29 kbs are packaged, which are not going to form viable plaques, viable phages. Okay?
Yes, sir. Got it, sir.
Yes. Good. Any other questions? Yes, please. We have 5 more minutes. I was looking at this syllabus, I think, somewhere.